Welcome to NSTA Web Seminars, where you can find live, interactive learning at your desktop. Today's seminar is part of our NGSS series, and it is titled NGSS Core Ideas from Molecules to Organisms, Structures, and Processes. Our presenters this evening are Aaron Rogat and Barbara Hoog. My name is Sue Hokkanen, and I will be moderating today's program. We will also be joined by Ted Willard this evening. Jeff Lehman is online with us tonight to also provide technical support. I want to remind you to visit the NSTA Learning Center, your online portal for professional learning with over 11,300 resources for science educators. You'll find that over 3,900 of those resources are free, and you can add them to your library to access when it's convenient for you. You can organize your resources by bundling them into collections, or access thousands of other collections made by the NSTA and other teachers. I encourage you to join the conversations in the community forums, where you can discuss content and classroom issues with other teachers, and our online advisors are available to help you find the information you are looking for. And you can use our free tools to help you organize your own professional development and meet your professional development goals. And all of this is available at learningcenter.nsta.org. Now I'd like to introduce tonight's presenters. First we will have Ted Willard, who is the Director of the NGSS at NSTA with the National Science Teacher Association, Aaron Rogat, a research scientist at the Educational Testing Service, ETS, and Barbara Hoog, who is a clinical associate professor at the University of Illinois at Urbana-Champaign. And we will start tonight with Ted. So Ted, if you're ready, thank you.
Thanks very much, Sue. Welcome everybody. So happy to have you all here. Looking forward to start talking about the core ideas in the life sciences. As a former physics teacher, this is new ground for me to think about in terms of all of these pieces. But I'm going to give you a quick sort of background about the standards so we all start on the same page, as it were, or on the same slide or screen, I guess, in a digital world. So I hope you're all familiar with the Next Generation Science Standards in some way. I just want to note that start off with that 4 organizations were involved in working with the standards: the National Academies, Achieve, the American Association for the Advancement of Science, and my personal favorite, no bias there, NSTA. And that there were 2 steps in developing the standards. There was a framework for K-12 science education that the academies were in charge of developing, and that was released in July 2011, and then the standards themselves. And from these standards, assessment, curriculum, instruction, and teacher professional development all derive from those standards. So let's start off with the framework and getting a little sense of that. What's important to know about the framework is really that there are 3 dimensions to the standards. There are science and engineering practices, cross-cutting concepts, and disciplinary core ideas. I hope everyone who's watching tonight has read the framework. There's really no excuse to not have looked at it. You can get a free copy of it, a PDF, from the National Academies Press. You also can purchase a copy from NSTA Press at the NSTA store. So to go and read that. If you read it once, you want to read it again, or at least read certain parts of it, are really useful in thinking about this whole section. And so looking at those 3 dimensions here, the first piece was the science and engineering practices, things like asking questions, developing models, analyzing data, constructing explanations. These are things that we expect students to be able to do in their classroom. And it is not just a method of learning, it is an outcome in and of itself. We want students, when they graduate from school, when they go into the world, to be able to do these things, to be able to think and act like a scientist in ways about going about their lives. Then next are the cross-cutting concepts, things like pattern, cause and effect, systems, structure and function. All of these things are scientific concepts, but they're not unique to any one discipline. They are found in life science and in neuroscience and in physical science. And so they play a role in all of those pieces, and it's why they're known as cross-cutting concepts. And finally, which is part of the story for tonight is the disciplinary core ideas. This is what traditionally people are familiar with, the content aspect of science. And so we have life science, physical science, earth and space science, and something new really in NGSS in many ways, at least for a lot of teachers and states, is engineering and technology. And each of these sections is broken down into farther, smaller details. Tonight we're looking at the first aspect of life science, from molecules to organism structures and processes. And each of those sections can be broken down further, and you'll see later on today that we'll go into the different pieces here in Barbara and Aaron's discussions. So all of that was described in the framework, and the folks who developed the standards went from that. And so an organization called Achieve sort of moderated or worked together with a group of 26 states. So all the states in blue here were part of that process. They were sort of a committee that really decided what the standards would be like. This was all a state process. There was no federal government involvement, for example, in terms of this. And each of these states had a review committee that would go through and look at different drafts of standards and provide feedback throughout the entire process. The standards themselves were written by a group of 41 writers spread across the country. Each one of those little dots represents one of the writers, so you can get a sort of a sense of the geographic spread that they all had. And about half of those folks were classroom teachers at the time where they did the standards. And some of them have moved on to other places outside of the classroom, but that to give you a sense that teachers were definitely involved in this process in a lot of ways. Here's a current situation with the standards. The states in green here have adopted the standards. States in yellow have done some sort of process. We should give a shout out. It's really hard to see, but the District of Columbia, just before the holidays, was adopted. So there's 8 states and the District of Columbia have adopted so far, and more are expected on the way. So here's an example of the standards. One piece of it, one of these performance expectations. And an important point about the performance expectations, they're describing what's to be assessed. They are describing what students should be able to do at the end of instruction. They are not, I repeat, not meant to say you as a teacher need to do this in your classroom. You still have total freedom to decide how you will go about instruction of these standards. But as we look at it, this performance expectation really pulls all the different elements from these 3 pieces underneath it. So let's go and take a look at this. We've got develop molecular models of reactants and products to support the explanation that atoms and therefore mass are conserved in a chemical reaction. That first section, developing molecular models to support an explanation, developing models, is one of the practices. The next element here, that reactants and products, the idea that reactants and products and that atoms and therefore mass are conserved in a chemical reaction. That's described here in the disciplinary core ideas. And then when it comes to looking at this idea of conservation and of energy and matter, that matter is conserved because atoms are conserved. That's a cross-cutting concept. And so this one performance expectation provides a way to assess whether students have gained an understanding of all these different elements in the standards. And so therefore, with that sort of background, I'm sort of understanding how the standards are constructed, these 4 different elements, performance expectations, practices, core ideas, and cross-cutting concepts. Gives us a little bit of place here that we can now really focus on some core ideas in the area of structure and function. So I'm going to pass that on to Barbara to share her pieces. Off to you, Barbara.
Thank you, Ted, and thank you everyone for joining in tonight. I know that a number of you have very busy teaching schedules, and I know a vast majority of you are either under extreme cold or expecting a storm tonight, and I truly do appreciate you all coming. I also wanted to add that I would actually like this presentation to be as informal as possible. I realize that it's a webinar and the interactions sometimes aren't necessarily as informal as give and take, but please do make use of the room that Sue pointed out earlier, and feel free to ask questions, raise hands, things of that sort. Both Aaron and I will ask for comment and feedback throughout the session, and to actually have your valuable input would be extremely useful for us. I am actually concerned that we've planned too much, but we'll see what happens. I have faith that Sue and Ted will keep us on track, but if we can't finish, I'm sure that we'll actually continue the discussion online. I hope that you're all willing to participate there or here. How it works out. So the next slide is just actually an introduction of who we actually are. And what I would actually like to do is just briefly turn it over to Aaron so that he can check his sound and to actually give a brief introduction of who he is. Aaron, would you like to talk?
Aaron, you may need to press the talk button.
Yes, sorry. Can you hear me now?
Yep.
Okay, great. Sorry about the crazy start for me. Those who are panicking as I try to get started here. Yes, I was trained as a cell biologist. Transitioned into K-12 science education. I'm currently at ETS. My current interests are developing innovative science assessments and promoting the use of learning progressions to inform assessment and instruction. In general, I'm interested in developing tools and resources to support the teaching and learning of science.
Okay, and I'll take it back over from Aaron now. As you can see, I received my doctorate in developmental biology from the University of Utah. And after a short postdoc at the Genetic Science Learning Center, an organization that many of you might have heard about, I actually transitioned permanently into science education. And I'm currently now down here in Champaign, Urbana, where I'm a faculty member in the College of Education. And here in the college, I teach in both the elementary and secondary teacher education. My primary interest outside of teacher education is actually in the design of curriculum and the implementation and design as well of professional development opportunities that can support teachers in the teaching of the challenging science that we would actually all like students to be learning. And I just want to give a plug for one of the projects that I'm currently working on, Project Neuron. In part, I want to mention this project because I think it has direct relevance to some of the content that we'll be talking about today and some of the things that we're trying to get teachers and students to be able to do and have the opportunity to do in this classroom. So if anyone is interested in learning more about Project Neuron, please don't hesitate to actually give me a call. But before we actually start and get into the content of our presentation, both Aaron and I would actually like to know a little bit more about you so that we have a better understanding of who our audience actually is. And so Sue, I'm actually going to probably ask for some help in this one. But what I would actually like people to do is to use the check marks or to use the code that I see that people are actually using already, the clicker, and actually select the answer that best fits your teaching about structures.
Okay, so underneath your name at the top, if you were not here for the introduction and you're not familiar with the Blackboard platform, underneath your name at the top you should see a letter A. Mouse over that and a drop-down menu should drop down for you to select your answer. And the question is, how much instructional time do you devote to teaching about structures? A, almost none. B, 1 or 2 lessons. C, 1 unit. D, I infuse it throughout the year. And again, if you're on a mobile device, you may not be able to respond to this question in the poll format. So please just go ahead and type your response into the chat area. And as I tell my new Science Teacher Academy participants, I'm from Chicago, where we vote early, vote often. So please let your voices be heard. This helps our presenters. And it looks as if we have a pretty strong representation. Barbara, if you're ready, I can lock these polls and publish these results for you.
I am ready.
Okay. There you go. I'll move it around so that you can read your responses too.
So I'm actually pleased to see that that the majority of people actually that I'm actually a little puzzled by the response in that a number of you have selected that you don't teach about it at all, D, and that could be that people haven't responded, or that the none means that it actually is actually, well, actually, can someone tell me why people don't teach structures in the time in their class?
Barbara, what you're looking at, the very last line, is actually participants that did not respond.
My mistake.
It says none.
Okay,
yeah,
my mistake.
Make sure that's clear for you.
My mistake. I'm actually glad to see that, because now the poll actually makes sense. So a number of you actually do teach it in many ways across your curriculum. And so hopefully as part of this conversation, people will see why this might be relevant to the new next generation science standards and to the framework. So let's see if I can actually do a little bit better in understanding how the check marks work in this next slide. And so what I would actually like you to do now on this slide is to use your either your clip art or a check mark or a pencil line and actually indicate here how you actually teach processes in your curriculum. And it's important here to think about both the production of food in plants as well as the use of food in animals, not necessarily at the cellular or molecular level.
I was going to step in with a reminder on where to find these, but I think they found them just fine.
Very good. And what people can actually see from this response is that a lot of you, most of you, all of you, teach at least 1, 2, 3, or perhaps all of these ideas in your curriculum at some level. And so this really just shows the importance of both structures in the previous slide and the processes here in teaching biology. And it begins to point out that you, in fact, are already teaching some of the content that actually is in the next generation science standards and in the framework more specifically. Okay, so thank you for giving that information and actually allowing me to learn a little bit about the poll system in the Blackboard system. What I'd actually like to do now is just briefly outline the structure of today's webinar that Aaron and I are going to be giving. And it's really divided into 3 main parts. The first is where we'll talk about the life science disciplinary core ideas and give a general overview of the what and the why of this discipline. And then we'll actually dive down into one particular core idea, from molecules to organisms, structures, and processes. And here we'll actually look at what is in this core idea. Aaron will actually talk about a possible learning progression. And then we'll actually shift to talking more about the teaching and learning of this core idea. The last bullet point, discussion, should actually be at a higher level because that's how we would actually like to end up the webinar, with a discussion where to provide an opportunity for you to ask questions, give comments, as well as feedback on what you think this idea is actually covering. So what is actually meant by a disciplinary core idea? So here on this slide, what I've actually done is simply identify 4 main points or 4 main bullets that can be thought of describing what a disciplinary core idea actually is. So as Ted introduced, and as you all know from reading the framework and looking at the next generation science standards, the new standards in the framework emphasize 3 main dimensions: the disciplinary core ideas, the practices, and cross-cutting concepts. And here tonight we're focusing on the disciplinary core idea. So the rationale for actually using this idea of the disciplinary core idea comes from looking at how scientists and engineers actually engage in their own work. Experts understand how core or foundational principles of their discipline and actually use these ideas to construct new understandings. They make sense of new research findings in their daily work or design solutions to problems that might come up if they're actually engineers. So if we actually want students to be able to understand the discipline, why not organize around these same disciplinary core ideas? And by doing so, hopefully we're setting the students up to develop the deeper understanding that will actually allow them to continue in learn science. Additionally, as pointed out here, a core idea needs to be thought of as a key organizing concept, either within a discipline or across multiple disciplines. Disciplinary ideas are foundational to understanding the discipline, and they allow one to explain a variety of phenomena. For example, if one understands the role of a cell structure or an organism structures in a particular concept, one can then perhaps apply that understanding in another situation. If, for example, looking at other cell types or other organisms that share some commonalities. Another example could be looking at the idea of cell theory to explain how plants grow, and then actually apply the same idea to understanding growth in animals or differentiation. In addition to be foundational, the ideas are often described as being generative to the discipline. If one learns the idea at one level, it can be applied to another to help explain a new phenomena or to allow a deeper explanation to be developed. The framework, when one reads it or rereads it, actually describes core ideas as providing a key tool for understanding or investigating more complex ideas. In addition, these core ideas also should be thought of as relevant across all grades and meaningful for the students. Using these ideas, it will become possible to actually engage students through connections to their own lives or connections to the larger society for an extended period of time. So why is it that the framework is focusing on the disciplinary core idea? Some of these ideas have already been mentioned in the previous slide in explaining what a disciplinary core idea actually is. But here we actually wanted to take a more student-focused approach and to stress why this idea might be helpful for students in learning challenging science. Core ideas allow students to develop a coherent and working understanding of the discipline. If we give students the opportunity to work with these ideas, hopefully they will establish a working understanding of what these ideas mean and how they connect with and across the discipline. It's important to think about how these disciplinary core ideas might help provide a context which can be used to engage the students to learn these core ideas. If the disciplinary core ideas become a focus of instruction, students should be able to explain a variety of natural phenomena, some of which might actually be phenomena that they haven't actually seen. For as much as we would like, we can't provide all the opportunities for students to see everything that they will ever see. But if we actually provide them with the ability to apply what they know and what they've done to new situations, hopefully they'll be able to explain and investigate new situations in new contexts. potentially contexts that we don't even yet know about. If we actually focus instruction around core ideas, hopefully students' explanations will become more and more sophisticated as they learn more and more about this idea. In part because it will be a foundational body of knowledge that they're learning across many years of instruction and use and ability to use that instruction and knowledge in many different ways. And finally, by taking this approach, one can hope that students develop an integrated understanding of all science, and now thinking about engineering, including engineering in this description, by having the core ideas structured so that they're teachable across multiple grades. Students will be able to develop the deeper, more integrated understandings that we all want students to develop. Hopefully, this will also help retain interest for students in science and engineering past the high school, or at least through the high school, and into their personal lives, in many cases, into their careers. So this provides a very brief overview of what and why we're focusing on core ideas. But I actually want to encourage people to read or reread the framework, chapter 2, where the authors actually talk about the 3 dimensions, in particular on page 30 and 31, where they give the justification for the disciplinary core ideas and how they identify what a core idea might actually be. So, if we want to teach this way and have our students learn science to this level, what exactly is in the life science core ideas that have been identified as core? And so here on this slide, I've simply listed the 4 disciplinary core ideas that can be found in the framework chapter on the life science discipline. Today, we're only going to focus on this first one, from molecules to organisms, structures and processes. And these 3 other ones have their own webinars scheduled for later this year. And I want to encourage people to actually go to those as well. So if we focus on LS1, from molecules to organisms, structures and processes, what is it that we actually find? What we actually find is that each of the core ideas are actually made up of component ideas. There are 4 component ideas within this particular core idea. Here, I actually list 2 of them, and on the next slide, I actually list the remaining 2. So, structure and function is what LS1A is. Growth and development of organisms is LS1B. And what you can see, I've listed 3 bullet points under each of these areas, where I pulled out information that you can read more about in the framework that make up the structure and function component idea. So this is the structure function, growth and development component idea, and then here is what makes up the last 2, LS1C, organization of matter and energy flow in organisms, and then LS1D, information processing. Again, I want to reiterate and encourage people to go back into the framework and actually read what is actually said about each of these different ideas. As you read over the points on this slide and think back to what was on the previous slide, you'll notice that these ideas are ideas that have been found in previous standards. They've been found in the National Science Education Standards, by the benchmarks, or the different state science standards that we've all been using. I don't think that there's anything here that might surprise people. Having said that, I do want to emphasize that not all of the information is presented on these 2 slides. So now that you've actually had a very brief overview, I want to turn it back over to you and get another poll. I want to have you answer this question. What do you think is the overlap between the National Science Education Standards, or perhaps other standards that you're more familiar with, and the Next Generation Science Standards when looking at the specific ideas present in molecules to organisms. Go ahead and take a few minutes and actually click on what you actually think.
Okay, and if you're just joining us and not familiar with the poll buttons are, if you look near the top, underneath your name at the very top of the screen, you should see a letter A. If you mouse over that letter A, a drop-down menu should appear. Just select your answer from that drop-down menu. A for none, B a little, C some, D almost all, and E for all. And Barbara, as soon as you're ready, just let me know and I'll lock and publish the results for you.
I'm sorry, why don't you go ahead and do it now?
Okay.
In the interest of time.
Okay, and I'll move this around so that you can still see the question, and actually I can enlarge it just a little bit too. There you go.
Good, good. Good. When Aaron and I thought about this question and were answering the question, we actually kept flip-flopping between C, D, and thinking more actually E. And then what we actually decided was, what we actually wondered was if the correct answer should actually be A, because here we're actually doing what the writers of the Next Generation Science Standards have told us not to do, and that we're disassociating content from the practices and cross-cutting concepts. And so if we actually think about it in that sense, there would actually be no overlap. But what we're actually thinking about in terms of content, I would have to agree, there would be either C or D with a possibility of E based on what you're actually thinking about in terms of overlap between the different standards. So if we actually do a comparison in looking at the NSES in the framework, we were wondering, what is it that we would actually find? Before we show you what we came up with when we did this, we would actually like 2 or 3 brave volunteers to volunteer to do a mapping of what they think the actual overlap is. So in this figure, or in this slide, what we actually have are 2 tables. The one on the left is simply taking the NSES, National Science Education Standards, grade band headings that are connected to the content that's in molecules to organisms. And on the right, we actually have the different component ideas taken from the core idea LS1. And what I would actually like 3 or 4 volunteers to do is to be given the power of the pen and draw lines that connect the left columns to the right columns in the rows that you think is actually appropriate.
Okay, so what?
And so
I'm sorry. What I'm going to do is, and Erica, if you are volunteering, you can leave your hand up. There you go. Leave your hand up, and then I'm going to take the next 2 or 3 that volunteer. Raise your hand, and that is the actual hand button next to the away button. And I'm only going to give whiteboard tools to a few. Okay, got 2. I need 2 more. Okay, 1 more. Okay, excellent. Very good. Thank you, guys. Let me go ahead, and I'm going to give whiteboard tools. If you're on a mobile device, you will probably not be able to use this. So I'm going all the way up through Amber is who I see here. Okay, all of you should have your whiteboard tools now. If you don't, then please let me know. Deborah, if you cannot see the slide, it could be that you're on a wireless device and you may need to adjust. Your connection speed. Go ahead and use your pen tool. And actually, I think what I'll do is there you go, someone knows what they're doing, to make the connections there. And I'll let Barbara speak to this part. But use your pen tool for what it is she's asking you to do.
You are doing exactly what I would actually like you to do. And to remember that you can, in fact, draw more than one connection from one cell to the other cell. Okay, is everyone done? Or is there any more lines to be drawn? Okay, why don't we go ahead and actually talk about what you've just drawn, and then also I'll show you what Aaron and I came up with when we did this ourselves earlier when we were planning the webinar. And what you can see here by the complexity of the numbers of arrows or lines drawn from one column to another column is that it's not a simple one-to-one correlation between the different areas from the old NSES to the new framework ideas. And in fact, there are many ways that these lines can be drawn and that actually show the overlap between the two ideas, the two different standards. Here on this slide is simply a representation that Aaron and I actually came up with when we were doing this drawing. And what you can actually see is that for some of these, there are many arrows that actually emanate out of one of the headings from the NSES to many of the different component ideas. And in fact, the characteristics of organisms, we actually have one arrow drawn for each of these ideas, whereas in a few of these, we actually have single arrows. The behavior of organisms arrow, for example, is drawn only to the LS1D information processing, whereas to give another example of many arrows coming out of one row is actually the cell heading from NSES, and that again goes to all of the different component ideas. One thing that I should point out is the fact that even though there might be many arrows coming out or lines coming out from one of the NSES grade band headings going into the different component ideas, not all of the lines necessarily have the same emphasis, so that's actually not shown here in this representation. This is just a different representation of looking at what the similarities are. So in the previous slide, you actually saw that you could draw lines that actually connect the old standards to the new standards. And in here, we've actually just pulled out some of the concepts that actually showed up on that previous slide. This is not meant to be an exhaustive list, but you can actually see that some of the core ideas, cells, structure, function, for example, are in both of the old standards and in the new standards. But perhaps more interesting and more importantly is what are the differences. So one key difference that I don't actually have here on this slide is the integration of the discipline practices and cross-cutting concepts. And so this approach is one key difference between the old standards and the new standards. What I do have here, though, listed on this slide and on the next, is actually some other key differences, some of which have already come up in the presentation. One key idea is actually the presentation of the discipline, in that you actually are thinking about things now in regards to having core ideas, not necessarily separate, disparate facts and concepts. And for this talk, in this webinar, focusing in on the molecules of organism ideas. And then, secondly, a key difference is actually breaking these ideas into a few coherent component ideas that actually give organizing structure to this overarching idea. Another difference that's actually key to thinking about the new standards is something that hasn't explicitly been talked about, is actually the emphasis between the connections between the core and the component ideas. All of the component ideas together actually make up the core idea. In addition, there are fundamental ideas that cut across component ideas. For example, the idea of cell theory. This idea is key in several of the component ideas. Mention of cells and roles that they play can be found throughout all of the component ideas. Cells is not simply something that is learned in one unit and then forgotten, or in one grade and then never mentioned again. Rather, students are expected to actually develop a working understanding of what cells are and how cells function at many different levels. And so this is another example of how an idea like this can actually provide an explanatory power for how organisms are structured and how they actually function. Similarly, students are being asked to develop an understanding of what structure and function is across multiple component ideas. For example, this idea comes up in both LS1A and LS1D, and it comes up across multiple levels of scale and organization. So it's not simply enough for students to learn about cells as a single entity, but rather the role of cells at the organism level as well, in many different levels in between. This new depth of understanding is also seen at the level of systems and system models for the ideas found within this particular core idea. Students are being asked to work with the ideas that systems are made up of parts, and that each system can, in fact, be made up of other systems. Interestingly, both of these ideas, structure, function, and system models, are actually cross-cutting concepts, showing how ideas within the core idea are actually linked to one of the foundational dimensions that the framework is actually structured around. So at this point in the session, what we would actually like to do is see if there are any questions, and either Aaron, Ted, or I can actually answer.
Yes, great. Thanks, Barbara. And to folks, if you've got some questions, just type them into the chat box, and then I will try to share them with Aaron and Barbara. Yeah, I did have one that came up. One point looked at your summary of some of the categories and noticed that the word molecule didn't appear in your bullets, and was curious about that. I'm assuming that the idea of molecules is used within the biology, with this section in the biology standards. And can you tell me when students are supposed to think about things in terms of molecules?
Yes, so that actually, molecules should have been there. I mean, in part, looking at the title of this core idea, it obviously is something that's important. And if you actually look at the LS1C, where you actually look at energy, the flow of energy, molecules certainly are mentioned there. And one of the key points that I would say about the molecules bit is that students need to develop an understanding of molecules that perhaps they learn in a more traditional chemistry classroom, and actually begin to apply it and understand what it actually means within a biological sense. So this integration of disciplines is something that's extremely important in the new framework. And that the term molecules, or the ideas beneath or underneath molecules, could actually be a perfect example of that integration. And I think that when Aaron actually talks about the learning progressions, molecules will in fact come up in different levels of there as well. Great.
Well, why don't we go on to Aaron's part here and hear about the learning progression. We can do some more questions after that. And Aaron, I think you need to push your mic button, turn it on again. Aaron, can you check and see if you've got your microphone on? I think Aaron's doing a microphone check here. So just hold on for a second. Aaron, feel free to interrupt me if and when you want. And please interrupt once you've got your mic working again. Let's see if we can get that. There we go. There we go, Aaron. Go for it.
All right. Yes, I'm going to go. We're transitioning now to talk about learning progression. So first I want to ask a couple of questions about learning progressions. And I'd like to take a poll here, if possible, yes or no. The first question, who here has heard about a learning progression, or the term, yes or no? Can you have a poll?
Yeah. Aaron, what I've done is I have placed a check mark where there used to be a letter A. There's now a check mark. So if participants can say yes, they can hit the green check. And if it's a no, the red X.
Okay. Okay, so yeah, it's looking like we have definitely, people have heard the term. Okay. So yeah, okay, it's good. A good chunk of people have heard it. I'm going to ask a couple of brave volunteers to just briefly tell me, or the others here, what you've heard about learning progressions. Just briefly, what you. It doesn't matter if you're still. It's not. You don't have to come up with a well-deformed understanding, and just briefly what you think you've heard about learning progressions. Can we get a couple volunteers for that?
Folks are just writing it in the chat room here. We've got Elaine, who said concepts that increase in complexity over the grade bands. And Barbara, if you're confused, we're asking right now for people to sort of give a definition of what they understood about learning progressions, what their understanding is it. And we've got Mary writing dovetailing of concepts over multiple grades. Heather noting how learning a concept is built through the grades. And Rachel, very honestly, and I appreciate it, not much at all. Now we're getting a lot of answers about concepts building and complexity over each grade, scaffolding through the grade levels, something that allows students to learn concepts from grade level to grade level, building on their knowledge, not just reviewing concepts year to year. So there's some answers for you, Aaron.
All right, thanks. Actually, that's certainly, those ideas are certainly appropriate. This idea of building understanding over time, and I think that is definitely relevant. I think what's key to the learning progression sort of thinking is that it really does take time for students to develop an understanding of these concepts, and we can't expect them to learn it. Cell theory, for example, over one unit of instruction. It takes time, so that's definitely one reason why we care about learning progressions. And so, and also, briefly, what maybe are you still unclear about? Can we get a couple of notes on that in the text box?
So I have people writing right now, so we'll just wait to have people share what they've thought. I can see that some folks are putting some things in. Michael's here has asked about whether it should include constructivism. How should we adjust our grading to assess learning progressions? And whether learning progressions are constructivism. Barry's got an interesting point here. I wonder about the coordination of teachers within a school, how they know depth from level to the next without teacher communication. And what pedagogical approach can be used to help learners progress towards a learning progression. I like Erica's acknowledgement of that she doesn't really know what she doesn't know.
Okay, well, I think that's pretty good. We have a range of responses here, from I don't really know to people thinking about constructivism, which is a theory of learning. I will say that is the idea that kids are building knowledge, integrating new ideas into their existing ideas is definitely part of learning progressions. There's a lot of questions around. Seems like a couple people were asking questions about how teachers use these and how they access them. And I think we're still, to be honest, we're still exploring these ideas right now. I thought one question that might come up is there really one progression for all students? And that's probably the one that comes up the most. And we typically say most people realize that there's students can take multiple pathways to get from their naive understandings to the sophisticated understandings, and some people are trying to characterize that, but there is an understanding that there are probably some important intermediate understandings that we need to understand and continue to get to in order to build new understandings. So I'm going to move forward now a little bit. Okay. All right. So this first slide, I'll try to whip through really quickly, is actually a definition, our definition of a learning progression. And we can come back to these questions people are having right now at the end of the end of this section on learning progressions too, and not at the end of the discussion as well. There's a lot of interesting questions going on right now being written. But quickly, we view a learning progression based as a empirically grounded and testable hypothesis about how students' understanding and ability to use knowledge and skill in core school subjects develop and become more sophisticated over time. Now I read this because it comes from a report that I contributed to, and I want to emphasize a couple of things about this definition. First, these are empirically grounded, testable hypotheses. They're theories of how students come to understand particular ideas or practices. And that's important because we put out these theories and they need to be tested. We hope that they're based on research. Ideally they are, and tested based on real results from students. But they do lay out a pathway to think about how students actually move from their naive understandings, which students have many when they explore a topic when in school, and how they move from those naive understandings, develop new understandings until they get to that point where they have the kind of understanding that's listed in those standards documents, that high school standards that experts believe are central to being proficient in that particular domain. And that's what society cares about. But it takes time for kids to develop those understandings. And by having a pathway of how students move through from their naive understandings to the most sophisticated understandings, we can have that as a roadmap to really think about instruction and curriculum and assessment to diagnose where students are along that pathway. That's the hope. I also want to note that, like I said before, in the definition, they should be based on research and they should be tested empirically. So the progression we're going to show here is based on some existing research, pieces of research on how kids think about some of these component ideas. It's not necessarily coherent. It's not following kids across time. We actually have to go out and test the progression ourselves, but it's still based on what other research has found about kids by interviewing kids and finding out what they know about some of these important ideas. And so I'm going to now shift to our hypothesized learning progressions, which needs to be tested, but is based on a review of some existing research. And what I'm going to show you is some text about each level in the progression, moving from the naive levels, the level ones, the early levels are always the most naive, the levels, in progressions, the higher levels are the most sophisticated, talk about the thinking at each level, and then have a little model here to illustrate some of the kinds of thinking that we predict. And I should note that this progression, we believe, or at least I believe, can be applied to all the component ideas from molecules to organisms. So whether it's information processing or structure function or flow of matter and energy, we believe this kind of thinking applies. And that's why we're talking about this progression in more general terms. So the level 1, which could be essentially at any level, it could be middle school, or you could find them at middle school or elementary or even high school in some cases, but likely you're going to find them early elementary school. These students tend to focus on macroscopic descriptions of organisms and their functions because they focus on what they can easily see and touch and feel, and that's how they think about organisms. They don't typically think about microscopic structure or details or behaviors or mechanisms at all. So they may not correctly associate specific functions with specific parts. When it comes to animal functions, they tend to anthropomorphize things. So they think about what organisms want as opposed to their physiological needs. So this characterizes level 1. And you would expect kids in early elementary school to have these kinds of views or thinking like this. So below here, you see a picture of a model of odor detection, which could be related to information processing, a phenomena related to that component idea. If you ask a kid to think about how could you smell a banana, how is it possible, you might get something like this from a level 1 student. You see the banana. I'm going to try to use my thing here. You see the banana. You see the nose. That's a macroscopic structure that kids easily see and think about. You see some streams here. The banana smell, representing sort of this cartoony kind of thing of smell, even though it's microscopic. And the nose, and not really thinking about a brain, the internal parts processing the information. This is typical of a level 1 student. Moving to level 2, which would likely be upper elementary school, you see a couple of changes in the model and in thinking. So they might begin to think about internal structures and organs more at this point, but they're still unclear about what they do inside, what the mechanisms are. So, for example, they might think about digestion, food digestion. They know somehow there's something going on inside of organisms and they digest things, but they just don't know what exactly that is. There's more focus on macroscopic structures and behaviors. Not so much microscopic structures at this point, so really not the cellular level. A little bit of rudimentary mechanisms, but not a lot. They are beginning to identify systems of a part in an organ system, so maybe the brain, for example, or the heart, a few parts, but not the whole system. So if you go to this model here, continuing this model of odor detection, a level 2 student, again, thinking about how to detect a banana, they would be somewhat macroscopic here. You see the smell, the nose, but maybe they think about the brain a little bit. That's some internal organ. They're not sure exactly where the processing is involved. Maybe they misassociate processing in the nose as opposed to the brain. They're not quite sure how the brain's involved. So there's a little bit of shift. There's some internal mechanism here, but not still a lot of pieces missing. Level 3, which would be early middle school, would represent the following thinking. They begin to develop explanations at the tissue and cellular level, but they still may make some errors. They can think more about macroscopic and microscopic structures, but they may inappropriately connect the two levels, the macroscopic and microscopic, or the easily visible and structures that are not so easily seen at the cellular level. They're still likely to focus on structures first and less about functions or behaviors or mechanisms. So in this model here, continuing to think about what level 3 would look like, see the banana and the smell again, but maybe there's some cells that they're beginning to think about in the nose that somehow they think the cells in the nose are involved. Still some missing pieces about how that's connected to the brain. Moving on to level 4 in this hypothesized learning progression, you begin to see the incorporation of cellular mechanisms into their models and explanations for organism functions, and more consistently, and that's important, so across more phenomena, and beginning to develop molecular level mechanisms. So we're talking about this is likely to happen in the end of middle school, when they're developing understanding of matter, and so they should be developing more molecular level explanations. They're making additional connections between more levels of organization. So now, if you think about it, you have macroscopic, cellular levels, and molecular levels. They have to be making those connections. Still, there may be more focus on function, associating those with structures, but they may not be able to move between these fluidly. And so if we get to our model of odor detection, again, you see the banana here, no longer the smell. You see some particles. They may believe these are visible. They can't be seen with the eye, which would be more appropriate for kids thinking about particles. And somehow are detected by special cell structures that have specialized parts to detect the odor particles. They may believe that understand there's some cellular connection between the nose and the brain. They understand the brain itself is also composed of cells. You can see, again, some more elaboration of cellular and molecular components in this model. And now moving to level 5 in this hypothesized learning progression, to be high school likely, certainly. Continued elaborate more molecular description and details in the explanations, a refined view of the molecular explanations, and consistently applied connections between the multiple levels of organization. And they can articulate more of the molecular behavior or mechanism and can move fluidly between these ideas. So in the model here, I should point out, and I know that Barbara and I have talked about this a little bit. This is the upper level. Students may not get to level 5 very easily. They may be stuck at a level 4, and may not refine their molecular explanations. But this is where a proficient student should be. And that's where we need to try to get them to into high school. And in terms of the level of detail here, you see banana again. If we continue on with the model, specific structures of molecules that are specific to the banana smell that are detected in the nose. Again, the specialized cellular structures that detect those and respond and bind to those molecules, like a lock and key analogy. The cellular connections to the brain, they understand there may be cell-cell communication at synapses, which also have some neurotransmitters, some molecular details there might be included. They understand there are specific regions of the brain devoted to specific functions, like a memory and a smell region. So you can, and actually you see some of this language in the the NRC framework that talks about this component idea. So more focus on, so more a complete system, more molecular details, more attention to function. So this, again, we believe this so this is just an example of one phenomena in the information processing where you can apply this progression, but you could apply it to growth and development. You could apply it to the flow of matter and energy in organisms. So we think that this is helpful in anticipating how students can think at different places in time throughout their K-12 career, where they need to be, what kinds of supports we could be giving them. So now I want to take a moment to ask questions about learning progressions in general. There are certainly a lot of questions that were being asked, or more specifically about this.
What we're suggesting to do, we've got about 10 minutes left in the program tonight, so we're thinking that if we could go on, let you go through the remainder of your slides here, and then we'll do some questions at the end here, so we can sort of try to stay in the neighborhood of being on schedule. All right?
Yes, thank you. Thank you for keeping us on track. Then I will move forward. Okay, so we have thought about supporting the learning of this core idea of from molecules to organisms, and a couple of suggestions we have in these slides. First couple slides are directly related to the progression I just talked about, because it anticipates challenges and supports that we're going to have to give students. So, for example, at early elementary school, curriculum instruction should focus on macroscopic descriptions of organisms and their functions. We know the students are going to be starting with macroscopic descriptions. We should take advantage of that. Learning progressions try to identify both achievements in thinking and the gaps in thinking. And so here's definitely an achievement. We take advantage of the fact that they can describe organisms pretty well, young kids can. And they may not be thinking about functions, but we can certainly have a focus on functions, reminding them about what the function is, ensuring that they understand the appropriate functions of these structures. And then we can ask for explanations of models, which we anticipate lacking some function and some mechanism. So we could support through instruction and curriculum that students think about maybe some rudimentary mechanisms, even if they're not clear on what those are, to get them to think about functions and mechanism. At upper elementary school, the progression suggests that students should be familiar with some organs to think about those internal mechanisms and their functions. So curriculum and instruction could focus on particular organs or organ systems that are relevant and make sure that students can begin to think about mechanism and another level of organization and systems. And so we have to be supporting students in thinking about those mechanisms, those unseen mechanisms, reminding them in curriculum instruction, think about mechanisms, even those that are unseen, because they're likely not to think about those without support. At the middle school, we know that they should be developing molecular and cellular level explanations in their models and their explanations. We should look for those. That kind of detail in their explanations. We are likely to need to support students in coming up with those explanations and to make sure that they are thinking about cellular and molecular level explanations. If they're not, they should be thinking that as criteria that they themselves should begin to apply when they look at others' explanations. Do they have cells? Is there a mechanism there that they're trying to articulate? Curriculum instruction should also help students make connections across multiple levels. By this middle school, they're thinking about, certainly by late middle school, they have to think about the macroscopic, the cellular, and the molecular, and they're going to need help making those connections. And we can ensure that through instruction and curriculum that they do that. Okay. High school, we're going to continue to require them to make multiple connections across multiple levels. I'll talk about the use of multiple representations at the end, but we're going to have to support this as well. It's a challenge. Now, quickly, a couple of problematic, naive conceptions that we believe teachers should anticipate finding and then trying to address, such as the failure to conceptualize organisms as made of cells or certain parts made of cells. People have found this to be a problem. Thinking about the difference between atoms, molecules, and even cells. This may be the result of students really not understanding, having sophisticated notions of matter, or their sense of scale is way off, and we have to support students in thinking about those relationships. Students may just they may think that living and non-living things are fundamentally different. It's hard for them to think about those cells made of atoms and molecules, even though it's being presented to them in instruction. Chemical reactions, even though there are a lot of processes involved in them, they get instruction around it, they still think of chemical reactions as something to learn in physics or chemistry, and not in life science. So we have to help them make conceptual change when they think about living systems. They don't attend to functional processes very much, and they often do not understand the purpose and function of things like mitosis and meiosis, because instruction and curriculum is focused on the details of these cellular processes and not the functions. And the same for processes like cellular respiration and photosynthesis. Ways to address these naive conceptions? Well, we believe thinking about the practices are great vehicles to do this. Constructing and revising models, getting kids to draw models, use physical models, use simulations, interactive media, forces them to think about their existing ideas, consider new evidence, and have to revise those. This has been done for some systems like the respiratory system and the circulatory system, and even an elbow at grade 2. Kids learn about a model elbow and can think about function with appropriate instruction, just as well as the 4th or 5th grader. So we know the models can support learning these ideas. We should also reflect the fact that scientists use models to think with. They're not just descriptions of phenomena. They test and revise their ideas. We want students to do the same thing when they create models. There's so many cases where students create a model of the cell, but they don't do anything. They don't think about it. They don't make predictions or explain phenomena with that model. We want students to be doing that if they're going to engage in modeling. I want to just put in a plug here. Modeling is also a great assessment tool. In addition to their text, you can have students draw and you get a rich insight into what they think. You can ask kids to draw at different points in instruction and can see how their mental conceptions are changing over time or not changing. There are examples of curriculum materials that actually engage kids in using models in this content area, and we have a couple links here. Another important practice is the practice of constructing and revising explanations and using evidence. We recommend thinking about evidence as cell biologists would, pictures or actual observations of organisms under the microscope, using videos of processes. This is what cell biologists do. They look at the structures and they think about their functions and processes. We know that there are slides out there often, but they're a narrow set of images. Local schools could work with local scientists and get perhaps a wider collection of images and slides to really collect evidence for some of these ideas in this core idea. Maybe even pairing real-world evidence with theoretical evidence, so investigations, for example, dissections to explore systems, theoretical evidence through interactive media, could be combined with that. Again, to ask kids to compare this evidence to their existing explanations or models and ask them to revise them. There are materials that ask students to construct explanations, and we have one here. And really, okay, how am I doing, Ted?
I'm sorry, Aaron. Ready for me?
How much time do I have left?
Just a couple minutes.
Okay. All right. Okay, and then real quickly, two last things to think about. We encourage the use of multiple representations for this particular core idea. There's two ways to think about it. Using multiple representations to explore a phenomena or its data set in different ways. So, for example, you can look at tables and graphs, different ways to look at the same data. You can look at a structure like a protein molecule using different representations, 3D models and 2D drawn models. Give students all these different representations, give students another way, another view to look at the data or the phenomena. And that can help them to find something new they didn't see from another representation. That's important to think about. Because there are multiple levels students have to think about, using multiple linked representations that show each level like the same thing, the macroscopic, the cellular, the molecular levels can be helpful to students. And then we have a couple examples real quickly of what this looks like, or what it can look like. Concord Consortium has done some stuff in genetics where they have dragons here, the macroscopic level of the dragon, specific traits. They talk about here, talking about meiosis and the distribution of chromosomes and genes, so they have a cellular level representation, chromosomes and even genes, specific allele. These can help students make connections. They're reminded about making connections. Another example here is Sim Scientist, which is really a form of assessment, but they are making, here they're representing the same phenomenon in different ways. This is blood and starch being taken into the gut, which is connected to the blood circulatory system, the blood system. And they're asking students to think about what happens when blood, glucose versus starch or complex sugar being transported into the cells. They make predictions about what will happen to the glucose and the starches, the complex sugars. They will see that glucose rapidly gets into the bloodstream while starch does not. They have representations, the pictures here to describe what's going on, and the data here to describe what happens to the glucose. So both of these examples are rich examples of how to use multiple representations. And then finally, we suggest don't forget about the cross-cutting concepts. They're very useful to help support understanding, to help the students to build those explanations and models that we're looking for. Specifically, systems and system models can be great to help students think about and reason about this kind of phenomenon in this core idea. Certainly thinking about the components of a system, their function in the system, the inputs and the outputs of the system. What happens when one part of the system is disrupted? How does that affect the rest of the system? Structure and function. These are cross-cutting concepts, and it's clearly pulled out in this as a component idea, in this core idea. The idea that certain structures have specific functions, they behave in a certain way. Patterns in qualitative data or quantitative data. Students should anticipate there being cyclical patterns to detect. There could be patterns at different levels in the system, the microscopic, the molecular level patterns, and the cellular patterns, and the macroscopic patterns. At different levels, there are different behaviors to be observed, to be anticipated. Scale, again, important. Students to be aware that there are things too small or too large to see. We need models to represent those. Often they have to be aware of the relative scale of these entities and develop an understanding of scale. These are opportunities are great. These can be taught in the context of the core ideas. You could be teaching systems and systems model as a unit, but then incorporate this core idea into it to help teach it. So we suggest definitely thinking about the cross-cutting concept to help support the learning. Okay.
So what we're going to do here now is I'm going to give a few pieces of information about some material that NSTA has. We'll then let folks fill out their review form, and then we'll come back and do some questions for folks. So let's just go through and let you know that all the discussions we've been answering some of your questions for you online here tonight, but the conversation can continue either we've got two websites of great importance. The standards are found at nextgenscience.org. Also, the NGSS at NSTA Hub, you can find at that address. We have a member-only listserv focused specifically on NGSS and a discussion forum, so we can continue the conversations we've been having in the chat at these places as well. I'd be happy to get information to Aaron and Barbara on how to communicate through there as well. We've got upcoming web seminars on biology topics you can see coming through here. The next 3 component ideas, and engineering and nature of science will be coming later in the spring. Lots of web seminars, all archived, our practices, cross-cutting concepts, core ideas, and assessment. We did have a big piece on assessment just a couple weeks ago. Also articles in all of our journals. I really encourage you to look through there. We're really trying to support the implementation of NGSS. A big piece here we're very excited about on Saturday, March 8th. We're going to do a one-day online conference, a virtual conference. Some of the people who've been involved in some of our web seminars will be doing this. It'll be a day-long event. Really encourage you to look there for more information about that. I'm very, very excited about this piece here, about looking at the practices, which is really the central element of the NGSS. The famed books, we've talked about the framework and the standards. I want to make sure you both know that there are reader's guides that NSTA has developed for both books. You can get at the NSTA bookstore. Also, Roger Bybee developed Translating NGSS for Classroom Instruction. So it's designed to help you move from the standards into what you do in your classroom. Roger is I think considered a minor deity in the life sciences and was on the head of the life science team. So if you're interested in that topic area in particular, it could be useful. But it's meant to be an example across all grades and things. There's an app for that. That's true for NGSS 2, either an Android device or iPhones. Your iPads, you can find the standards there. And I hope to see all of you at our conference in Boston. You could do nothing but do sessions on NGSS if you were there. There'll be, in particular, a one-day event on Saturday at the conference, April 5th, that will be a series of sessions around NGSS. So keep an eye out for more information about that. They'll be free to all conference participants. We've also got PDIs that are focused on the standards as well. It's the place to be to learn about NGSS and all this. And with that, I will thank again Aaron and Barbara, who did a great job getting into the life sciences here. I think we've had some great information about learning progressions, about the standards. And with that, I will pass things off to Sue to do her little bits of magic.
Thanks, Ted. And please, let's give a round of applause to all of our presenters. And again, the smiley face. You're going to mouse over the smiley face, and from there, pick the applause, and you'll actually see your hands clapping underneath your name. Excellent. Thank you, guys. And again, thank you to Ted, Aaron, and Barbara for an excellent web seminar this evening. Thank you very much for sharing your evening with us and giving us this valuable information with the NGS standards being adopted in many states and being considered in others. And I'd also like to thank the Carnegie Foundation for supporting today's program. A thank you to the administration of NSTA for their support of web seminars. And you can Find resources related to today's presentation by going to the web seminar collection in the Learning Center, which will include the archive to this web seminar and the presentation slides. The collection may also include journal articles and lesson plans, interactive content modules called Science Objects and SciPacks, and links to helpful websites and multimedia. I'm going to put a link directly into the chat window for this collection, and I've just done that. So please bookmark that for later. Also, everyone who's registered for this program will receive an email within the next day or so with a link to access the archive and collection. And here's a look at what's coming up on the web seminar calendar. On January 29th, we have Engineering Design Process on the Moon. On January 30th, we have Enhance and Extend Your Professional Learning with the NSTA Learning Center. And on February 4th, Engineering Design Challenge, Water Filtration. You can register for upcoming programs by visiting the learningcenter.nsta.org/webseminars. And I am actually going to post a link very quickly for you in the chat area also so that you can bookmark that for later. There you go. Okay, so thank you everyone tonight for your participation. We look forward to seeing you again on another NSTA web seminar soon.